Hello student, in this video I am going to discuss about staining method. So what is meant by staining, how it is importance in the microorganism. The coloring agent is important, a color to the colorless microorganism. So due to this coloration the microorganism become visible. So as to observe its cell shape and structure. So these stains are composed of positive and a negative ion, one of which is colored and is known as chromophore. So the basic dyes are using positive ion while acidic dyes in the negative. So since bacteria are towards negatively changed at pH 7.0, so thus the colored positive ion in a basic dye binds to the negatively charged bacterial cells. So some of the dyes are classified acidic dye and some are classified as a basic dye. So what are the different type of staining method? The first, negative staining. So in the negative staining, as stated that earlier, that acidic dye due to the negatively charged bacterial surface does not interact with the dye negative ions, so the background stain color. So this type of preparation of where the colorless bacteria are visible against a colored background, so is called negative stain. What are the dyes which are used during the negative stain? Eosin, nigrosin, Indian ink, these etc. These are negative stain. Next, simple staining. A aqueous or alcoholic solution of single basic dye is used called simple staining. For example, methylene blue, crystal violet, saffronin. So sometimes a chemical helps to intensify the stain. Such a chemical is called mordant. So the mordant is closely associated with the stain and shows its affinity. So in the case of stain is applied to fixed smear, then washed off, slide kept of drying, and then it examined under the microscope. So such a procedure helps in studying the cellular shape and structure of microorganism. Next method is differential staining. It is the most important. Differential stain is used in bacteriology. Is Gram stain, which was discovered by Danish bacteriologist Hans Christian Gram. So it is the only procedure based on staining technique which divide bacteria into two large groups, that are gram positive and gram negative. So the procedure of staining is, since the crystal violet important purple to the cells is called a primary stain. So the purple dye after washing off from smear is to be covered with iodine, that is mordant. Both type of bacteria appear dark violet or purple in color. So smear then washed off ethanol or ethanol acetone solution which act as a decolorizing agents. So remove the purple's color from the cells, some of the species. Thus the ethanol is rinsed off and again stained with saffronin. So the basic dye which act as a counter stain, washed again and blotted dry and examined under microscope. So what is the mechanism of staining? So it is believed that mechanism means the staining depends upon the structural difference in the cell walls of the gram positive and gram negative bacteria. This is confirmed on the basis of cell wall of gram bacteria is removed and treated with gram stain. The bacteria turn gram negative. Thus the cell wall of the gram positive consists of a single 20 to 80 nanometer homogeneous petroglycan or murine layer lying outside the membranes. So while the gram negative cell wall has 1 to 3 nanometer thick peptidoglycan layer surrounded by 7 to 8 nanometer thick outer membrane. So during the gram staining technique, the bacterial cell size appear to be about the two-thirds length of the cell before staining. So hence it causes the degree of